Another really important concept that we need to understand from muscles that also helps us introduce some of the things going on with the neurons and our neural system, our nervous system, is this thing called a neuromuscular junction, which, it's the joining or where the neurons, the cells of the nervous system, and the muscle cells come together. We already know that intuitively that our muscles can be paralyzed if something happens to the nerve. So what is it that the neuron is actually doing for the muscle? That's what we want to look at right now. Okay, here we actually see a fascicle. Remember, a fascicle was a group of muscle cells, or also called muscle fibers, together. But we need to somehow get the information from this is actually the spinal cord going out to a spinal nerve. And then this is one cell, one neuron that's branching where it's dendrites and cell bodies. Remember, cell bodies are always in gray matter area. So here we actually have the cell body that's embedded up here in this area of the spinal cord, this anterior wing of the gray matter here, and the axon though is actually protruding out through the ventral side. It's called the ventral root, and then going into a spinal nerve. And then this nerve is actually an organ, but this axon is part of a cell called a neuron. Anyway, when this neuron goes out towards where it's going to release its neurotransmitter, then it may have several different branching areas. And in this case, we see these different branching areas that are going to allow a signal called an action potential. You may call it a nerve impulse, and that is another name for it, but we'll mainly be calling it action potential. And it's going to trigger somehow to release a neurotransmitter. Then something the neurotransmitter is going to do is going to help us elicit a contraction. So let's look and see what that is. Okay, neurons that actually go to muscles are called motor neurons. Neurons that actually would go back here and then go up and be interpreted in the parietal lobe would be sensory neurons. So since we're talking about muscles and the neurons that innervate them, then we're talking about motor neurons. What we're really interested though in this topic is where the actual neuron comes to a junction next to the muscle. They don't actually make contact, but they're extremely close together, and there's some chemical things that are going to happen there that we're going to talk about. Now, just to orient ourselves, this is the neuron, this is the end of the neuron, the axon, where the axon is terminating. So this is the bulb of the axon. So here's the axon bulb. These blue areas are actually vesicles full of neurotransmitters. In this case, the neurotransmitter is acetylcholine. There are different neurotransmitters that are released by neurons. We'll be talking about some of the different ones later. But the ones that are released across to muscles is the neurotransmitter called acetylcholine. Now, let's look at the muscle that we have here, because here's our sarcolemma, that outside membrane, that cell membrane. And then we see that we actually have this three-part section here. This is the T-tubule. This part is part of the triad. Here's the terminal cisternae. Here's the terminal cisternae. These little red dots in here are actually calcium. So we also have a folded area of the membrane. It's actually called a motor end plate. And then we have a space between the synaptic bulb and the motor end plate, and that space is the synaptic cleft. Okay, what else do we have here? Well, here we have our Z line with our thin filaments coming off of it. Here we have the thick filaments. So this space between the end of that thin filament and that thin filament, this would be the H zone through here that we talked about earlier. So here we actually, here's a little bit, we can see of the other Z line, which of course would be anchoring these other thin filaments, the actin. All right, now, but what we want to talk about is what's going to happen here. What's actually happening? How is the relationship between this going to trigger this to go through a contraction? Okay, again, here we see all the parts that are tagged. You do need to be familiar with all these different parts. And again, notice the axon terminal, the end of the axon, where it actually has like an enlarged bulb-like area. You have this space here between So this is not literal, this is actually not making contact with the muscle, but instead it's coming extremely close to it, but does have a little bit of a space, which is necessary because we have to have a space to release our neurotransmitter into. Okay, so here again is our neurotransmitter, acetylcholine, that is going to actually be in the synaptic vesicles. Our motor proteins actually move the synaptic vesicles, fuse them with the end of the membrane to release and cause exocytosis of the neurotransmitter to happen. Now, again, the motor end plate is really special. Again, it's a phospholipid bilayer membrane, just like the sarcolemma is and like the ends of the T-tubule are. But there's a difference. There is a different type of a doorway that is actually in this membrane. There's a different doorway in this membrane than there is in this membrane and this membrane. The doorway here, I'm going to call it a keyed doorway. The technical word for it, it's a chemically gated channel. Now, the neurotransmitter doesn't actually go into the muscle cell, but the neurotransmitter is the key that opens up the door that allows ions to flow into this from the outside of the cells into the cell. Because as we've tried to build a little bit of an understanding, there's always more sodium outside of cells than inside of cells, so we'll always want to try to go in. There's always more potassium inside of cells than outside, so we'll always try to go out. But it can't just go through that phospholipid membrane. It can't do simple diffusion. It has to go through some sort of doorway. So one of the keys to how this works is understanding the doorways that are in both the motor end plate, the sarcolemma, and the T-tubule, and the doorways that actually exist in the terminal cisternae. Now, one of the most important concepts that you really have to understand, and if you get it now, it'll be easy to continue to add to the information as we talk about the neurons themselves, but is this thing that we see here, what's called a polarized condition. As we know, water was a covalent polar molecule, meaning that it was one molecule that had positive charges on the hydrogens and a negative charge on the oxygen. Well, now we see both in this cell and in this cell, notice that we literally have a negative charge just inside the membrane and a positive charge just outside the membrane. There's a difference in the ion concentration on either side of the membrane. Therefore, it creates a charge or a voltage difference, which we literally can measure. Now, notice we have, again, the positive on the outside, negative on the inside of no matter if we're talking about the neuron or we're talking about the muscle cell. And that's really based primarily on the fact that we always have more sodium, a positively charged ion outside, and it's always wanting to try to go inside through those doorways. Now, this charge doesn't necessarily exist further inside the cell, but it does exist across that membrane. Therefore, we call it a resting membrane. Again, that's the membrane, the phospholipid bilayer, whether we give a specific name to it like a motor end plate or like a sarcolemma, but that membrane has a potential, in other words, an ability for the charge to change, especially if we get an influx or an efflux of ions either into the cell or out of the cell. So when we are starting out, the resting membrane potential is always negative if we measure it, and we literally measure it in millivolts. So right now we just need to understand that there's a negative charge just inside the membrane compared to a positive charge on the outside of the membrane when nothing else is actually happening. And then when we actually get our impulse, what's happening in our impulse, it's literally we're having positive ions rush in, and then we will see that our polarization will do the opposite. For just a millisecond, some very brief time period, the inside will become positive and the outside will be negative in relationship to it. But then, and when that happens, we say we depolarized the cell. But then when potassium rushes back out and it becomes negative back inside versus the positive ions outside, then we say that is repolarization. So we're going to be talking about the resting membrane potential. We're going to talk about depolarization, meaning we change the polarized condition. and repolarization, meaning that we move this condition back to its original, how it started, which is always negative on the inside and positive on the outside. But again, notice that exists because
although plus and minus signs are,
because this is inside the cell, and even right here is technically outside the cell. It's outside the membrane, but it's always the difference in the charge across the membrane. So again, when nothing's really happening, when the impulse or the action potential is not moving through here, then if we measured this, we would come up with a specific millivoltage number that we call the resting membrane potential. Okay, next we actually see a sequence of events that's going to occur. First, the action potential, which again is the nerve impulse, is going to arrive. We will notice that because we'll see the negative change to positive, positive change to negative, and it will do it in little sequences. It won't do it all at one time, because what's actually happening is there's little doorways here that sodium is moving through, and so when sodium moves through those, when those doorways open and sodium moves through it, then that becomes positive and this becomes negative. Then when it changes back or repolarizes, then what happens is potassium ran back out. Now, again, we have this thing called the action potential, so we're going to see the signal move down through here. Then we're going to see depolarization of the motor end plate. Now, once what we're going to actually see that the signal moving down in here, the reason why it does, it actually, that signal opens up calcium voltage gates. Once we change our signal, we change our voltage, so it opens up calcium voltage gates. Calcium comes in, triggers my little guy, my little motor protein to grab the vesicle and to drag this to the end and release the neurotransmitter. Now the neurotransmitter acts like a key and opens up the next doorways, which then allow more ions to come in and change the charge on the inside of this as in relationship to the outside. That's called depolarization. Because if I am at negative on the inside and positive on the outside, and all of a sudden I become positive on the inside, negative, then I've changed my polarization. I've depolarized it. Okay, now, of course, following depolarization, repolarization will occur. But once we depolarize it, that signal then all of a sudden starts moving across the sarcolemma and down the T-tubule. All right, now, again, the reason why it's moving across and down is because now we start opening up voltage doorways, not the chemically gated or the keyed doorways. Okay, so I'm going to go ahead and play this little part because it's important you actually see what's happening. And I may pause it and explain a little bit of it.
voltage change of the membrane opens voltage-gated calcium channels, allowing calcium ions to enter the axon terminal. To see the arrival of the action potential, click the motor neuron. Click the calcium channel to see calcium ions enter the axon terminal.
Now look, what happened was we actually had the signal moving through here, the action potential or voltage, the action potential or the nerve impulse, I'm trying to say. Okay, now notice at this point right here, at this point and at this point, we are depolarizing that section right there, meaning that enough ions rushed through the doorways in the phospholipid bilayer to make that voltage rise to a positive number compared to what it is outside. Okay, now the real reason why we sent the action potential down here and changed the voltage is because we have doorways here that happen to be voltage gated calcium channels. In other words, these are doorways that will only allow calcium to come in to this portion of the cell if we change the voltage. So it's kind of like having an electric door and the electricity opens it up. So our signal's kind of like that electricity going through the door and opening up the door so the ions, the calcium that's outside the cell, can rush in. The calcium here actually then serves to trigger my little guy who pulls vesicles down to actually fuse them with the wall of the bottom of the axon so it can go through exocytosis and release the neurotransmitter. Calcium coming in actually triggers, if you remember this guy, which let's watch him walk there just for a second. Okay. calcium actually triggers him